Ebola kills roughly half the people it infects. Marburg can reach 88%. These numbers terrify us, and they should. But by the standards of deep evolutionary time, these viruses are not apex predators. They're clumsy amateurs, thrashing around in unfamiliar hosts, applying the wrong strategy to the wrong biology. The truly lethal viruses, the ones that achieved near total kill rates, existed billions of years ago, and they dominated life on Earth for longer than complex organisms have existed. They didn't negotiate. They didn't go dormant. They extracted everything and left nothing behind. Then, over hundreds of millions of years, they went extinct. Not because we defeated them, but because the physical world changed in ways that made their strategy obsolete. This is the story of why ancient viruses could afford to be 100% lethal, why modern viruses cannot, and how the most successful pathogens in history became permanent passengers written into your DNA. A virus that kills you in 3 days has failed at evolution. This seems backward. We think of deadly diseases as powerful, as apex threats, but lethality is not the same as evolutionary success. A virus is a biological machine with two functions: replicate inside a host and transmit to the next host. If it kills the host before transmission, the virus dies with the body. If it cripples the host so badly that the host cannot move or interact with others, the virus has quarantined itself. Natural selection does not reward violence. It rewards transmission. Epidemiologists describe this through the virulence transmission trade-off. Virulence means damage to the host. Symptoms, organ failure, death. Transmission means getting to a new host. The equation balances these forces. High virulence can work, but only if the transmission rate is also high. If a virus can jump to a new host instantly, it can afford to destroy the current one. If transmission requires time, mobility, or specific behaviors, the virus must keep the host functional. This trade-off explains why the deadliest viruses in modern history tend to burn out quickly. They kill too fast to spread efficiently. The ones that persist, herpes, Epstein-Barr, are the ones that learn to wait. But this logic only applies to complex, mobile life. For the first 3 billion years of life on Earth, the rules were different. The environment permitted maximum virulence because transmission was guaranteed. The ancient viruses that dominated the microbial world were not evolutionary failures. They were perfectly adapted to a world where lethality had no cost. 3.5 billion years ago, life existed in layers. Stromatolites, massive reef-like structures built by cyanobacteria, covered shallow seas. These were not individuals. They were dense colonies where millions of bacteria lived pressed together in mats of slime called biofilms. The extracellular matrix held them in place, neighbor to neighbor, generation after generation. For a bacteriophage, this was an all-you-can-eat buffet with no exit strategy required. Bacteriophages are viruses that infect bacteria. Their replication method was, and often still is, lysis. The virus injects its genetic material into the bacterial cell, hijacks the cell's machinery to build hundreds or thousands of copies of itself, then triggers the cell to explode. The bacterial membrane ruptures, the contents spill out, and the viral progeny scatter into the surrounding water. In a biofilm, they scatter directly onto the next victim, which is physically glued to the one that just burst. The transmission rate was effectively 100%. There was no penalty for killing the host because the next host was already within reach. This strategy is called rapacious parasitism. The virus extracts every available resource from the host. It does not leave energy for the host's survival. It does not modulate its replication to extend the host's lifespan. It takes everything, destroys the cell, and moves on. This worked for billions of years because the density of life made it work. The bacteria could not flee. They could not develop complex behaviors to avoid infection. They could not mount an immune response that remembered specific viruses. Every encounter was first contact, and first contact was almost always fatal. The lack of adaptive immunity meant viruses did not need to be subtle. Bacteria had innate defenses, cell walls, antimicrobial peptides, and CRISPR-Cas systems that acted as primitive memory by storing fragments of viral DNA. When a bacteriophage infected a cell, the CRISPR system could cut out a piece of the viral genome and insert it into the bacterium's own DNA as a record. If that same virus attacked again, the system recognized the stored sequence and destroyed the invader before infection could proceed. This was the closest thing to immune memory that existed in the ancient world. But viruses adapted. They evolved anti-CRISPR proteins, molecular tools that disabled the bacterial defense system. A virus would inject its DNA along with anti-CRISPR proteins that bound to the CRISPR machinery and shut it down, rendering the bacterial memory useless. The bacteria then evolved new CRISPR variants. The viruses evolved new anti-CRISPR counters. This arms race ran faster and more brutally than anything seen in complex life. Both sides were single-celled, with generation times measured in hours or minutes. Mutations that provided even a slight advantage spread through populations within days. This speed came at a cost. High mutation rates destabilize genomes. Many mutations are neutral or harmful to the virus itself. But in a world where the alternative was extinction, viruses that mutated rapidly had a chance to survive. Those rapid mutations often broke cellular machinery in messy, violent ways. A virus that could breach a new bacterial defense by scrambling its surface proteins didn't need to do so elegantly. It just needed to get inside and replicate before the cell died. The result was high virulence, not as a strategy, but as a byproduct of the evolutionary velocity required to stay ahead. The Red Queen ran at sprint pace, and nothing about the race rewarded restraint. When life became multicellular and mobile, distance entered the equation. A fish infected with a parasite cannot transmit that parasite to another fish unless the two fish come into contact. If the parasite kills the first fish within hours, the corpse sinks to the seafloor, and the parasite dies inside it. The next potential host swims past, uninfected. The rapacious strategy that worked in biofilms became an evolutionary dead end in open water. Mobility created the mobility barrier. A virus that wants to spread through a population of animals must keep its host alive and functional long enough for that host to encounter other hosts. This requires restraint. The virus must replicate, but not so aggressively that it destroys critical tissues. It must extract resources, but not so much that the host collapses. The optimal strategy shifted from maximum extraction to sustainable extraction. Viruses that caused chronic, low-grade infections outcompeted viruses that caused explosive, fatal infections. The structure of multicellular organisms added another layer of difficulty. In a single-celled bacterium, infection equals access to the entire organism. In a complex animal, the virus must navigate through layers of tissue. Skin, mucus, and epithelial barriers protect the internal organs. A virus that infects a skin cell and immediately lyses it creates a localized lesion and goes nowhere. The virus is now trapped in dead tissue on the surface of the body, with no access to the bloodstream, no route to the respiratory tract, and no way to reach another host. It has effectively quarantined itself. To succeed, the virus must move through the body without destroying every cell it touches. It must replicate in surface tissues slowly enough that the cells remain functional while the virus navigates deeper. It must reach the bloodstream or lymphatic system, where it can spread systemically and eventually exit through breath, saliva, blood, or other fluids. This journey takes time. A virus that kills surface cells instantly never completes the journey. A virus that replicates quietly, moving from cell to cell without triggering immediate necrosis, reaches transmission points. Natural selection favored the patient over the explosive. Multicellularity also introduced compartmentalization. Different tissues serve different functions, and some are more critical than others. A virus that replicates in the gut lining may cause diarrhea, but leave the host mobile. A virus that replicates in the heart or brain may kill the host before transmission occurs. Early viruses that could not distinguish between tissue types often infected whatever they encountered first. If that tissue was expendable, the infection might persist. If it was critical, the host died, and the virus with it. Over time, viruses evolved tropism, affinity for specific tissues. Targeting the respiratory tract, digestive tract, or blood allowed replication without immediate lethality. Targeting the heart or central nervous system did not. Social behavior created a final pressure. As animals began to live in groups, herds, packs, colonies, they developed instincts to avoid sick individuals. A wolf that shows visible signs of severe illness, lethargy, disorientation, visible lesions, is abandoned by the pack. The sick animal dies alone, and the virus dies with it. But a wolf that appears healthy while carrying an infection will continue to interact with the pack, hunt with the pack, and transmit the virus through shared kills or physical contact. This created selection pressure for the incubation period. A virus that kept the host asymptomatic for days or weeks could spread to dozens of new hosts. A virus that made the host visibly horrific within hours spread to none. Stealth became a survival trait, and the ancient apex killers that lacked it went extinct. 450 million years ago, jawed fish evolved adaptive immunity. This was the most significant shift in host pathogen dynamics since the origin of life itself. Adaptive immunity is not a wall, it is a library. When a virus infects a host, the immune system captures fragments of the virus, stores them, and builds specific antibodies to target that exact pathogen. If the same virus returns, the immune system recognizes it immediately and destroys it before it can establish an infection. For the first time in Earth's history, hosts could remember their enemies. This forced viruses to make a choice. They could remain loud and aggressive, replicating rapidly and causing visible symptoms, but this meant the host's immune system would catalog them and eliminate them on second contact. Or they could become quiet, replicating slowly, hiding in tissues where immune surveillance was weak, and persisting for years or decades. The viruses that chose stealth survived. The viruses that remained aggressive were purged. Latency became the dominant strategy. Herpes viruses, for example, infect a host and then go dormant inside nerve cells, where the immune system cannot easily reach them. They can reactivate later, but by that point, they have already transmitted to other hosts. HIV replicates slowly and targets the immune system itself, disabling the very cells that would destroy it. These are not accidents. They are the result of millions of years of selection pressure applied by adaptive immunity. The appearance of antibodies also created an evolutionary pressure for surface protein variability. A virus's surface proteins are what the immune system recognizes. If a virus can mutate those proteins quickly enough, it can evade the immune memory. This is why influenza requires a new vaccine each year. The virus is constantly reshuffling its surface markers. But this strategy comes with a cost. High mutation rates destabilize the virus's own genome, and many mutations are lethal to the virus itself. The evolutionary arms race did not make viruses stronger. It made them quieter, more evasive, and more patient. Before adaptive immunity, a virus could be messy. It could trigger massive inflammation, destroy tissue indiscriminately, and still succeed as long as transmission occurred. After adaptive immunity, that mess became a liability. The immune system would remember the inflammation, remember the tissue damage, and mount a faster, more aggressive response the next time. Viruses that survived the transition were the ones that learned to whisper. Viruses do not leave bones. Their protein shells decompose. Their genetic material degrades. We cannot dig up a 100 million year old virus and study it under a microscope, but we can read the scars they left behind. Roughly 8% of the human genome consists of endogenous retroviruses, ERVs. These are not fragments or remnants. They are complete or near complete viral genomes, permanently integrated into our DNA, inherited from parent to child across millions of years. Retroviruses replicate by inserting their genetic code into the host's DNA. Usually, this happens in somatic cells, lung tissue, blood cells, liver cells, and when the host dies, the virus dies with it. But occasionally, a retrovirus infects a germline cell, a sperm or an egg. If that cell is fertilized and develops into an organism, every cell in that organism's body will carry the viral DNA. If that organism reproduces, its offspring will inherit the virus. The infection becomes heritable. It becomes a fossil. Scientists use molecular clocks to date these fossils. DNA mutates at a relatively constant rate over time, a process called neutral drift. By comparing the same ERV sequence in humans and chimpanzees, researchers can calculate how long ago the two species shared a common ancestor and how long ago the viral infection occurred. Some ERVs are shared across all placental mammals, meaning the original infection happened more than 100 million years ago before those lineages diverged. More remarkably, scientists can reconstruct the proteins these ancient viruses produced. By reading the DNA sequence of an ERV, they can synthesize the viral envelope proteins and surface receptors in a lab and test how they behave. Early studies using this method revealed that ancient viral proteins were far more aggressive than their modern counterparts. They bound to host cells more readily. They triggered cell fusion at lower concentrations. They were optimized for rapid entry and rapid replication, consistent with the rapacious strategy that dominated the microbial world. The genomic record also reveals a shift. ERVs that integrated before the evolution of adaptive immunity show genetic structures optimized for speed and lysis. The viruses were changing in response to the hosts, and the hosts were changing in response to the viruses. The arms race is written into both genomes. Some ancient viruses did not just stop killing, they became essential. This is the strangest and most profound outcome of the virulence transmission trade-off. A virus that integrates into the germline is no longer just a parasite. It is part of the host's inheritance. If that viral gene happens to provide a useful function, natural selection will preserve it. Over millions of years, the virus becomes a gene. The parasite becomes a tool. Syncytin 1 is the clearest example. This protein is produced by a gene that was originally part of a retrovirus that infected a primate ancestor roughly 25 to 30 million years ago. The virus had a surface protein designed to fuse cells together, creating a large multinucleated cell that the virus could use as a replication factory. It also had mechanisms to suppress the host's immune response so the virus would not be attacked during replication. Both of these features were useful to the virus. They are now useful to us. Syncytin 1 enables the formation of the placenta. Specifically, it allows cells in the developing embryo to fuse into the syncytiotrophoblastic, the outer layer of the placenta that connects the fetus to the mother's blood supply. It also prevents the mother's immune system from attacking the fetus, which is genetically 50% foreign. Without this viral gene, placental mammals as we know them would not exist. Pregnancy as we experience it would not be possible. We are not just coexisting with this virus, we are built from it. ARC protein is another case. The ARC gene in the human brain produces a protein essential for long-term memory formation. When a neuron needs to send information to another neuron, it packages genetic material into ARC protein shells and transmits them across synapses. The protein structure is nearly identical to a retroviral capsid, the shell that holds viral RNA. The mechanism is viral. The function is cognitive. Our ability to remember this information is powered by a tool borrowed from an ancient infection. These are not isolated examples. The human genome contains thousands of ERVs, and while most are inactive or degraded, many have been co-opted for functions in placental development, immune regulation, and neural activity. Evolution did not just neutralize the threat, it repurposed the machinery. The most successful viruses in history are the ones that stopped being viruses and became us. If evolution trends toward coexistence, why do Ebola, Marburg, and rabies still kill? Why does avian influenza have a mortality rate above 50% in humans? These seem to contradict the entire trajectory. They look like throwbacks to the ancient world, apex killers that never signed the treaty. But they are not ancient. They are accidents. Ebola is a peaceful passenger in fruit bats. It has co-evolved with bat biology for thousands or millions of years. It replicates slowly enough that bats remain healthy and mobile. It does not trigger catastrophic immune responses. It transmits efficiently through bat colonies. The virus is adapted to bats. When it jumps to a human, it is a biological mismatch. The virus applies the replication strategy that works in bats to the human immune system, and the result is hemorrhagic fever, organ failure, and death within days. Ebola is not an apex killer. It is a clumsy tourist, breaking furniture because it does not know the layout of the house. This is true of nearly all modern viruses with high lethality. They are zoonotic spillovers, infections that jumped from their natural host to a new species that they have not had time to adapt to. Rabies evolved in carnivores. Marburg evolved in bats. Avian influenza evolved in waterfowl. When these viruses infect humans, they are operating in unfamiliar territory. They have not yet been shaped by the virulence transmission trade-off in the human context. Given enough time, evolution would tame them. But spillover events are rare enough, and human-to-human transmission is inefficient enough that the virus often dies out before it can adapt. We have watched this process happen in real time. In the 1950s, the myxoma virus was introduced to Australia to control rabbit populations that were destroying agricultural land. The virus originated in South American rabbits, where it caused mild symptoms. In European rabbits, which had no evolutionary history with the pathogen, it was catastrophic. Initial mortality rates reached 99.8%. Within a single season, millions of rabbits died. The virus seemed to be an unstoppable apex killer. But within a few years, the death rate began to drop. By the end of the first decade, it had fallen to roughly 70 to 90%, and it continued to decline. This was not because the rabbits evolved resistance, though some did. It was because the virus evolved lower virulence. Rabbits that died within days of infection transmitted the virus poorly. They became sick, retreated to their burrows, and died in isolation. Rabbits that survived for 2 or 3 weeks, those infected by a mutated, milder strain of the virus, remained active, continued to interact with other rabbits, and spread the milder strain to dozens of new hosts. The milder strain outcompeted the lethal strain, not because it was better at killing, but because it was better at spreading. The peace treaty was signed in real time, driven purely by natural selection. HIV is undergoing the same process now. It jumped from chimpanzees to humans roughly 100 years ago, crossing over through the butchering of bushmeat in Central Africa. In its early decades in the human population, it was highly lethal, killing most infected individuals within 10 years if untreated. But already, researchers have documented strains that progress more slowly, cause less severe immune damage, and allow longer survival. This is not due to treatment. Untreated individuals infected with certain strains show significantly delayed progression to AIDS compared to those infected with earlier, more aggressive strains. The virus is calibrating. Strains that kill their hosts too quickly are less likely to transmit. Strains that allow the host to remain asymptomatic for years transmit more effectively through sexual contact and vertical transmission from mother to child. We are watching the negotiation happen, generation by generation. The ancient apex killers, the ones that achieved near total lethality in their hosts, are extinct. They could not survive the transition to complex, mobile, immune-competent life. The modern killers are new arrivals, still learning the rules. They will either adapt or fade. The viruses that persist are the ones that have already learned to wait. The arc of viral evolution does not bend toward mercy. It bends toward transmission. Dead hosts are dead ends. Paralyzed hosts are quarantined. The virus that keeps you alive, mobile, and unaware is the virus that spreads. Natural selection does not care about your suffering, but it does care about your survival, at least long enough for the virus to reach the next person. This is why ancient viruses were so deadly. They existed in a world where proximity was guaranteed, where immune memory did not exist, and where mobility was irrelevant. Lysis worked. Rapacious extraction worked. Total lethality worked. The environment permitted it. When the environment changed, when life became multicellular, mobile, and immunologically sophisticated, the old strategies failed. The viruses that adapted survived. The ones that did not went extinct. The most successful viruses in Earth's history are not the ones that killed the most hosts. They are the ones who stopped killing and started negotiating. Some became chronic infections, persisting for decades without causing symptoms. Others went dormant, hiding in nerve cells and reactivating only when conditions were favorable. A few went further. They integrated so deeply into their host biology that they became indispensable. They became genes. They became us. The peace treaty was not signed in a laboratory or a treaty hall. It was signed in the structure of DNA, in the slow grind of natural selection, in the extinction of every virus that refused to compromise. We carry the victors inside every cell. We build our children from viral proteins. We remember the world through viral capsids. The war is over, but the terms were written by evolution, and they are permanent.